Hello, I'm Essay, your AI essayist. Please note that all content, audio and video in this presentation are AI generated. As AI can sometimes produce inaccuracies, I encourage you to verify any factual information presented. With that in mind, let's begin.
The mid-20th century presented a landscape of profound medical triumphs juxtaposed with enduring, terrifying biological enigmas. Among the most feared was poliomyelitis, a disease that cast a seasonal shadow of paralysis and death, symbolized by the chilling apparatus of the iron lung. The scientific community, for all its advances, remained fundamentally stymied. The prevailing dogma held that the poliovirus was obligately neurotropic, meaning its replication was believed to be confined exclusively to nerve cells. This single belief created a formidable barrier, as nerve tissue is notoriously difficult to maintain in culture, rendering larger-scale viral propagation for study and vaccine development an almost insurmountable challenge. It was within this climate of intense public fear and scientific impasse that a paper emerged from the Research Division of Infectious Diseases at the Children's Medical Center in Boston. The 1953 publication in Science by John Franklin Enders, Thomas H. Weller, and Frederick C. Robbins, titled Cultivation of the Poliomyelitis Viruses in Tissue Cultures, represented far more than a mere technical advance. It was a conceptual earthquake, a methodological key that unlocked the future of virology and irrevocably altered the course of public health. This work, born of meticulous observation and a willingness to question established doctrine, provided the essential practical foundation upon which the great polio vaccines, and indeed the entire edifice of modern viral cell culture, would be. The intellectual soil from which the Enders, Weller, and Robbins breakthrough grew was tilled by decades of preceding effort in the field of tissue culture. The very concept of keeping cells alive outside the body, or in vitro, was a frontier explored early in the century by pioneers like Ross Granville Harrison and, more famously, Alexis Carrel. Carrel's work, while groundbreaking, demonstrated the immense difficulty of maintaining organized tissues and preventing contamination over long periods. His techniques were demanding, an art form accessible to a select few. A subsequent and crucial step was taken by the British researchers H. B. Maitland and M. C. Maitland in the late 1990s. They developed a simpler method using minced hen kidney tissue suspended in a serum thyroid solution, allowing them to cultivate vaccinia virus. This Maitland culture was a significant simplification, moving from whole organ perfusion to using flasks of suspended tissue fragments. Yet, it was still a crude system. The cells were in a soup, though individual status was difficult to observe, and the method was ill-suited for the perceived delicacy of the poliovirus. The central problem persisted: how to grow poliovirus efficiently and visibly. The work of Charles Armstrong in 1939, who successfully adapted the Lansing strain of poliovirus to cotton rats and other rodents, offered a subtle crack in the neurotropic dogma, suggesting the virus might possess a wider cellular hospitality. It was this subtle hint, combined with the accumulated technical knowledge of cell culture, that set the stage for the Boston group. Their own prior success cultivating the mumps virus, a project on which Weller was leading, provided them with the direct experience and confidence in the roller tube culture technique, a method where culture tubes are placed on a slowly rotating drum to bathe the attached cells continuously in nutrient medium. The monumental contribution of Enders, Weller, and Robbins lies in its elegant subversion of the central dogma through a straightforward and reproducible methodology. Their research began with a deliberate and revolutionary choice of tissue. They procured human embryonic tissues, specifically skin muscle and intestine, obtained from therapeutic abortions. Their critical insight was to test whether the poliovirus would replicate in these non-neural cells. This was a direct challenge to the established belief system. The tissues were finely minced and anchored to the inner surface of glass test tubes using a plasma clot. To this, they added a nutrient medium, a carefully designed cocktail that included Hank's balanced salt solution, bovine amniotic nuclein fluid, and horse serum. Crucially, their medium also contained the antibiotics penicillin and streptomycin. This addition was a masterstroke of practicality, solving the persistent problem of bacterial contamination that had plagued earlier tissue culture efforts and often obscured or invalidated results. The prepared tubes were then placed in a roller drum, ensuring the cells were alternately exposed to the liquid medium and gaseous phase, optimizing their health and adherence into these meticulously prepared cultures of thriving, non-neural human cells. They introduced the poliovirus. They worked with all three known serotypes: the Lansing strain for type 2, the Brunhilde strain for type 1, and the Lyon strain for type 3. The team then simply watched. What they observed through the microscope was the core of their discovery, a phenomenon they termed the cytopathic effect, or CPE. Within a few days of inoculation, the cultured cells began to show distinct and progressive signs of degeneration. The normally elongated fibroblast cells would first show increased granularity in their cytoplasm. Then they would retract their processes, rounding up into dense, spherical forms. Ultimately, These degenerating cells would lyse, or burst, and detach from the glass surface, leaving behind empty patches in the cellular monolayer. This destructive process was observable directly under a standard light microscope. It was a clear, unambiguous, visual signature of viral activity. They rigorously confirmed their findings. Control cultures, which were uninoculated, showed no such degeneration. The cells remained healthy and confluent. To prove that this cytopathic effect was specifically caused by the poliovirus, they performed neutralization tests. They mixed the virus with polio-specific immune serum before adding it to the cultures. In these tubes, the cells remained healthy, the antibodies in the serum having neutralized the virus and prevented it from destroying the cells. This confirmed both the identity of the agent and the specificity of the effect. Finally, they took the fluid from the degenerated cultures and inoculated it into monkeys and mice, which subsequently developed the characteristic signs of paralytic poliomyelitis. This final step proved that the virus grown in their non-neural cell cultures was still virulent and fully capable of causing the disease. Closing the logical loop of their investigation, the immediate impact of this 1953 publication was seismic, resonating through the field of public health with unparalleled speed and force. The primary and most celebrated consequence was the direct enablement of polio vaccine development on an industrial scale. Jonas Salk, working at the University of Pittsburgh, had already formulated the concept for an inactivated or killed virus vaccine. His great obstacle was a matter of supply. The only existing source of poliovirus was the spinal cords of infected rhesus monkeys, a method that was astronomically expensive, ethically fraught, logistically nightmarish, and yielded vanishingly small amounts of virus heavily contaminated with nerve tissue, which itself could provoke dangerous autoimmune reactions in vaccinees. The Enders paper provided Salk with a missing piece of his puzzle. It was the manufacturing blueprint. Using their techniques, it became possible to produce vast quantities of poliovirus in large bottle cultures of monkey kidney cells, a tissue source that was more readily available and manageable than human embryonic tissue for mass production. The virus could then be harvested from the culture fluid, purified, and inactivated with formalin to create the Salk vaccine. The success of the 1954 field trial, the largest public health experiment in history, the direct and undeniable legacy of the Boston group. Simultaneously, the technique was equally vital for the work of Albert Sabin, who championed a live attenuated oral polio vaccine. Sabin's approach required a method to serially passage the virus through non-human cell cultures to select for viral variants that lost their neurovirulence while retaining their ability to provoke a strong immune response. The Enders method provided the perfect system for this process of attenuation. The cytopathic effect offered a clear visual readout of viral replication, allowing Sabin to quantify the virus at each stage and test its properties. He could meticulously cultivate, isolate, and characterize his attenuated strains, a process that would have been impossible using live animals alone. The subsequent global success of the Sabin oral vaccine in controlling and, in many regions, eradicating polio owes its existence to the foundational in vitro system that Enders, Weller, and Robbins had so elegantly demonstrated. Beyond the vaccines, the work revolutionized diagnostic virology. Before 1953, confirming a polio infection or determining a person's immune status required complex, expensive, and time-consuming procedures involving live monkeys. The new technique allowed for rapid and inexpensive in vitro neutralization tests. A patient's serum could be mixed with a standard dose of poliovirus and added to a cell culture. If the cells survived, it meant the patient had protective antibodies. This transformed polio diagnostics from a specialized research procedure into a routine clinical laboratory test, enabling larger-scale epidemiological studies and precise tracking of the disease and vaccine efficacy. The long-term legacy of the 1953 paper extended far beyond the conquest of polio. The methodology became the new gold standard for virology, a versatile template that could be adapted to isolate and study a host of other viral agents. Enders himself, along with Thomas Peebles, used the same fundamental technique to achieve the first isolation of the measles virus in 1954, another pivotal achievement that directly led to the development of a highly effective measles vaccine. The technique was subsequently applied to isolate and cultivate rubella, mumps, varicella zoster virus, adenoviruses, and countless other pathogens. It effectively opened the floodgates of viral discovery. The development of continuous cell lines, most notably the HeLa cell line derived from Henrietta Lacks cervical cancer cells in 1951, acted in perfect synergy with the Enders method. These immortal cell lines provided a consistent, endlessly reproducible, and standardized biological substrate for viral cultivation, further simplifying and scaling the process. The combination of the roller tube and, later, stationary monolayer culture technique with continuous cell lines became the workhorse of virology labs worldwide for decades, underpinning fundamental research into the viral life cycle, viral genetics, and the development of antiviral drugs. The paper's conceptual contribution was equally profound. By demonstrating that poliovirus could thrive in non-neural tissue, it shattered a limiting dogma and forced the field to reconsider the tissue tropism of all viruses, opening new avenues of investigation into pathogenesis and viral entry mechanisms. The reception of the work was almost immediate and universally laudatory. A rare event in the often contentious world of science, the importance of the breakthrough was self-evident. It solved a problem that had frustrated the world's best scientific minds for half a century. The most telling evidence of its impact was the remarkably swift awarding of the Nobel Prize in Physiology or Medicine in 1954 to Enders, Weller, and Robbins, a mere year after their definitive publication and 5 years after their initial findings were reported. The Nobel Committee recognized that the work was of a different order. It was a gateway discovery that enabled a whole new era of research and intervention. Critiques of the work itself are difficult to find, as its empirical basis was so solid and its utility so immediate and profound. The meticulous controls, the clear visual evidence of the cytopathic effect, and the confirmation of virulence in animals left little room for scientific doubt. Any initial skepticism about the ability of poliovirus to grow in non-neural tissue was quickly swept away by the overwhelming and reproducible evidence. A more modern, retrospective lens might introduce a note of historical irony. The team's use of human embryonic tissue, a matter of practical necessity at the time, was conducted in an ethical environment far simpler than today's. The procedure, so crucial to the initial discovery, would now be surrounded by complex regulatory and ethical debates. This historical observation highlights the shifting contextual frameworks within which science operates. Another subtle, almost philosophical, point is that the work was a triumph of observation over complex theory. It required no profound new law of nature, merely a clean experimental system and the wisdom to see what was in front of them, an approach that stands as a testament to the power of fundamental, curiosity-driven bench science. The influence of specific individuals and prior works on the Boston team is clear. They stood on the shoulders of giants like Carroll and the Maitlands, who pioneered the very possibility of keeping cells alive in vitro. They were inheritors of the practical knowledge of media formulation developed by figures like Raymond Parker and Morgan, and they directly benefited from the discovery of antibiotics by Alexander Fleming, which made their clean cultures possible. The direct influence of their work on others is a roll call of mid-century medical heroes. Jonas Salk and Albert Sabin are the most prominent, as their vaccine programs were entirely dependent on the viral production methods Enders, Weller, and Robbins had perfected. Thomas Peebles, working in Enders' own lab, directly applied the technique to isolate the measles virus. Wallace Rowe and Robert Hubner at the National Institutes of Health used similar methods to discover the adenoviruses. The paper became required reading, a foundational text for a new generation of virologists who no longer had to rely on the costly and cumbersome use of live animals. It democratized virology, making it accessible to any reasonably well-equipped laboratory. In conclusion, the 1953 paper by Enders, Weller, and Robbins represents a watershed moment in the history of medicine. It was a work of profound elegance, combining a simple but revolutionary conceptual shift with a robust and reproducible technical method. By liberating the poliovirus from its perceived prison within the nervous system, they provided the world with the means to see, to quantify, and to cultivate its feared adversary. This act of liberation led directly to the development of the vaccines that would transform polio from a terrifying annual scourge into a preventable disease on the verge of global eradication. Yet its legacy is even broader, for in solving the problem of polio, they provided a universal key that unlocked the study of countless other viruses, laying the essential groundwork for the golden age of virology. The paper transformed the human relationship with viral disease, shifting the balance from one of reactive fear to one of proactive understanding and control. It stands as an enduring monument to the power of challenging dogma, of meticulous observation, and of how a simple, elegant technique observed in a glass tube can echo through history, saving millions of lives and reshaping the possibilities of science.
We really appreciate you tuning into this AI-powered presentation. Our goal is to push the boundaries of AI in content creation, and your input is invaluable. If there's a particular subject you'd like to see us tackle next, or if you have suggestions on how we could fine-tune our AI's creative prompts, please don't hesitate to leave your ideas in the comments section. We're excited to bring you more unique content very soon.